It's a pleasure for me to be here. My first words, I think, are my words of compliments to the organizers, in particular Andrea. I have been at several occasions to meetings that Andrea has organized, and the standards and the setting the framework is actually unbeatable. So we cannot make such a course in Germany. So not by that standards. So I hope you all enjoyed yourself and you make new networks, connections. I was supposed to talk about cytometry of immunity. That is a big mouth title, yeah. So I of course have to somehow talk on specific subjects in that area. But up front, I would like to know actually who of you is actually working in immunology or is using lots of immunology approaches, is educated in immunology. Is that, can I? So, okay, nearly everybody. So I don't have to go into basics. So thank you very much. I think the challenge, actually cytometry really took off a little bit. Cytometry is an old science, but really the advent of fluorochromes and the ability to, and the advent of monoclonal antibodies made it a ménage à trois, which actually really led to a flourishing new kind of science. And the immune system itself and its reactions, I think, are a challenge. Oops. Are a challenge for cytometry because the immune system is everywhere in the body. It's guarding the cells, the skin surfaces. It has an own independent vessel system, the lymphatic vessel system, with the lymph nodes here as meeting places for reactive lymphocytes. But the cells are also in the blood. They are lining up at the gut. And as I will talk a little bit more in detail later on, they are also in the bone marrow. Actually, in adult humans and mice, mammals, they are originating in the bone marrow. And as I will try to convince you, they are also actually, the bone marrow is a preferred homing place for cells of the immune system. Now, when talking about what are ways actually to use cytometry in immunology, I will address today three different subjects, limit myself to that. The first one is ways to detect antigen-specific lymphocytes, and can we use that actually to monitor immune reactions, and in clinical terms, can we find ways, can we actually learn about the mechanisms of disease that have to do with the failure or hyperreaction of lymphocytes to certain antigens. The second topic I want to address is the tissue that is most accessible and most people use, and probably most of the talks here were actually focusing on cells from the blood. And I will actually address the question, is the blood really a representative tissue? Are all the lymphocytes or a fraction of all the lymphocytes in the blood, or do we have to take a little bit more care in interpreting the data that we obtain from the blood? And finally, I will very briefly elucidate a little bit on the other way around. Actually, if it is not a specific lymphocytes, but can we use the leukocytes in the blood as kind of biosensors for systemic changes of the immune system, for example, for chronic inflammatory diseases, and do these cells reflect, so to speak, kind of an imprinting of the body for certain types of diseases and the response to therapy. So I will first start with the detection of antigen-specific lymphocytes, and I will talk about work that was coordinated by Alexander Scheffold in our place and conducted mostly by Petra Bacher, former PhD student, now postdoc in Alexander's group. And actually, the point I will address is actually, can we somehow monitor the reaction of an individual's immune system to harmful versus harmless environmental challenges: parasites, viruses, bacteria, fungi, pollen, and food. And I will concentrate a little bit on one particular pathogen, which we will meet in a moment, which is a fungus. So, in principle, just one slide for those who are not so familiar with immunology is that one key feature of the immune system is that B and T lymphocytes have specific antigen receptors, specifically recognizing particular structures, but not any other structure. That's kind of the hallmark of them. Actually, I probably just as a matter of an anecdote, when I was a student, my mentor was Klaus Rajewski, and I once asked him about monocytes and macrophages, and he said, no, those cells don't have antigen receptors, they don't belong to the immune system. Yeah? So there was an old traditional definition of the immune system as you need antigen receptors. Yeah? I think nowadays we look at it a bit more relaxed, but still antigen receptors are a key feature of the immune system. And the B lymphocytes who have as an antigen receptor surface antibodies are a bit have a bit advantage because they recognize soluble antigen, and their receptors usually have very high affinities. Of course, there are also B lymphocytes with low affinity receptors, but the affinity can be up to 10 to the minus 12 molar. Now, T cells have antigen receptors which evolutionary are actually related to antibodies, but they are designed actually to recognize antigen in the context of antigen-presenting molecules presented by antigen-presenting cells. And that means that, that the affinity of the antigen receptor for monovalent antigen is much lower, orders of magnitude lower. It's actually just 10 to the minus 5, 10 to the minus 7 at most. But if the antigen comes actually as a cluster, an oligomer, for example, in the frame of a tetramer with 4 antigen-presenting cell peptide molecules, then the avidity, the combined affinities on the cell surface can be as high as that of antibodies to a soluble antigen. Now I know that other people here at this course have talked about oligomers that directly stain antigen-specific T cells or fluorescent antigen that directly can stain antigen-specific B cells. I will not go too much into detail with that. I will talk a little bit about indirect measures to look at antigen-specific T lymphocytes, and and that is according to measuring by cytometry their reaction. That could be either, in the case of B cells, the secretion of antibodies, or it could be actually without cytometry, isolating the cells and then allowing them in vitro to develop into antibody-secreting plasma cells and to detect the antibodies in the supernatant of these cells. For the T cells, actually, there is, for many years, different ways actually to assess the reaction of specific T lymphocytes to a certain antigen because they are activated to proliferate. They start to express molecules, co-stimulatory molecules with which they actually communicate with other cells. They secrete cytokines, also ways to control immune reactions, or they secrete actually cytotoxic molecules, ways to kill other cells. Now, in graphical terms, the specific T cell receptor recognition with peptide MHC multimers, as you have seen it in this course before, is kind of a trivial chemical way. Actually, one should say that also this way to detect specific T cells is not without caveats. For example, the question whether or not the T cell is activated by actually the staining reagents. Now, proliferation assays using brome desoxyuridine or other base analogs to label proliferating cells or fluorescent dyes which are diluted upon proliferation or cell division have quite a long tradition. And degranulation of cytotoxic cells and also cytokine-producing cells is not that familiar in the field, CD107A and B staining. And then there is the detection of activation markers, which either can be detected before they are secreted by intracellular immunofluorescence, or, as Hyun Dong probably has talked about already, after secretion using the affinity matrix technology that we developed about 20 years ago. Now, the advantage to use activation markers, for example, over the tetramers or oligomers is that actually you can use complex antigens, and you will assess all antigen-reactive CD4 T cells, or also CD8 T cells. Unlike for the oligomers or tetramers, where your detection limit is restricted to certain peptides. Even if you use banks of peptides, these may, in the best case, reflect a cross-section of what a real pathogen has to offer. So you can actually access all, not only antigen but pathogen-reactive T cells. The manipulation in vitro is limited to a brief re-stimulation if you go for memory-type cells. It takes a bit longer if you go for naive cells. And a further advantage is in clinical routine that these reactivation assays are independent of MHC restrictions, so independent of what MHC the patient has. As long as you stimulate the patient with the patient's own antigen-presenting cells, the cells will be restricted or will be able to activate or reactivate antigen-reactive T cells of a patient. Now, a critical point in this is, for the story that I'm going to tell first, is that the reactive markers of activated T cells are different for those T cells that actually stimulate an immune reaction and entertain it, the conventional T cells. And one of the best markers for this is CD154 or CD40 ligand. Or for those T cells that actually inhibit immune reactions, the regulatory T cells, because those T cells do not express CD40 ligand. Instead, they express a co-stimulatory molecule called 4-1BB or CD137. So, and that actually, so that was problem or characteristics number one of this reactive, antigen-reactive flow cytometry. Now there comes a second challenge, and that is the low frequency of these cells in the blood. At this time here, we limit ourselves to looking at these cells in the blood, and actually this is the frequencies of these cells. You see the highest frequencies against certain viruses like CMV, which are actually not really eliminated over time, is up to 1%. But many allergens, for example, have frequencies of as low as 10 to the minus 6. Autoantigens, the same thing, and also among naive cells. So the frequencies are between 1 in 10,000 and 1 in 1 million cells. That means actually that the numbers of cells that you will detect in a given amount of blood is limited. So we, many years ago, developed a technology called MAX technology, which you're probably all familiar with now, because the first author of this paper actually set up a company, so that's commercially available by now. And that actually the purpose of this development was to develop a parallel separation method. Parallel means all of the cells, the decision whether the cells are separated is made for all of the cells at the same time in a column, in a magnetic column. And that these magnetic particles that we use are so small that they don't interfere with flow cytometry, so that we can take these cells and immediately put them into a flow cytometer and detect them by flow cytometry. And we use very small superparamagnetic particles in the nanoscale range. Actually, it's an early example of nanotechnology, which are so small that in flow cytometry you need a lot of these beads on the surface of a cell just even to affect the scatter properties. So Alexander Scheffold now, since a few years, combined these two approaches by taking peripheral blood, adding antigen for a few hours, then labeling the cells with fluorescent and magnetic CD154 and CD137 antibodies, and then enriching the positive cells over a magnetic column. And you see here on these 4 dot plots, first of all, without magnetic enrichment, here the cells reacting to an antigen lysate, actually a lysate of a fungus, Aspergillus fumigatus. You will realize in a moment why we talk about Aspergillus in this particular case here. And you see here that if you do this restimulation in a control culture without the lysate, but everything else, and here you have the lysate in the culture, you see some positive cells here, very few. Actually, the total events, a number of events in this dot plot is 40 and 80 here, so very few cells, very limited fidelity. You also actually don't really know what the background is. You have actually here, without antigen, already 44 cells, and just 77 with the antigen, so you have 30 cells more. So it's not really very high discrimination. Now, if you do the magnetic pre-enrichment, then without antigen, you still have very few cells here. It's about the same order of magnitude as without magnetic enrichment. But you now see here prominent populations of more than 1,000 cells. And you see actually immediately that the cells expressing CD137 and the cells expressing CD154, the conventional T cells and the regulatory T cells, are actually completely separate populations. This is exclusive. There's very few cells which are here expressing or seem to express both markers, probably just variation. So you get these 2 cell types. Now, of course, so that you see actually, so just to put that into numbers, that the sensitivity for detection of antigen-specific cells is increased by about 1 order of magnitude if you, because you don't enrich cells, the background cells from the negative sample. What you enrich is the positive cells here. Now, this, the advantage of this is not only that you have a higher sensitivity, but also that you now can go, and I don't want to go into detail for this here, I just want to show you that if you now have thousands of cells, you're actually able to detect small subpopulations. For example, here, 25% of the cells against Candida albicans in this particular case here, or only 3% of the cells in the case of Aspergillus of this particular patient are expressing interleukin 17. So that is a small subpopulation, but it could have decisive functions in the controlling the immune reactions. So using this antigen reactive T cell enrichment procedure, you are able now to detect small subsets of reactive cells. So now about how do you control the specificity. I don't talk about this tetramers here, but in the functional assays, I showed you one control already, namely the restimulation cultures all minus antigen, like the FMO controls and conventional flow cytometry. And then I will show you 2 more controls, and that is actually the activation with other antigens and the block of antigen presentations. So here is shown in this particular case here, if you block MHC presentation, you see here without blocking, and here the effect of the blocking antibodies, highly significant reduction in the reaction to these antigens or pathogens here. Actually, one should talk more about pathogens than individual antigens. Or if we block antigen processing, as in this particular situation, you see here how efficiently you can get rid of the activation of these cells. Oops, sorry. And then the specificity control 2 is actually, can we, if we turn these cells into short-term T cell lines, can we show that they specifically react to the antigen that we used to isolate them? And this is a chart, actually, a table where we used the 5 different antigens: tetanus, adenovirus, CMV, Candida, so yeast, and Aspergillus. And you see here there's a perfect diagonal. So the cells reacting are re-reacting only to the antigen that was used to isolate them in the first place. So Aspergillus is a pathogen that we, all of us, probably encounter in our lifetime. And And most of us stay healthy, so we actually kind of are not reacting by allergy. So some of us are actually overreacting and becoming allergic to antigenic determinants of Aspergillus. But there's also an even worse, actually, an insufficient reaction, which is the invasive infection, which actually, in the end, destroys our lungs if it is not clinically treated. So can we use, actually, now cytometry, this RT technology, to find ways, actually, to, first of all, diagnose these clinical pathogenic situations, allergy versus invasive infection, and the healthy state of all of us. And second, can we learn by cytometry actually what is the mechanism of disease behind this overreactions? So the first thing is that the same picture I showed you actually already. This is a healthy individual here. And we now first have to show that these, first I want to convince you that these Tregs are really Tregs. So if we go for these cells, the red cells here are the CD137 or 41BB positive cells. Their phenotype is that they express CD25, that they express FOXP3. Most of them, 2/3 of them, also express Helios. And they express, some of them express CCR7, but all of them express CD45RO. They express CD137 among CD25. All of them express actually both markers. And what is actually a critical marker, we think, for the identification of Tregs, of true Tregs, is the demethylation of a particular control region in front of the FOXP3 gene, which actually imprints the cells to express FOXP3, the lineage determination markers of regulatory T cells. So much to the phenotype. If we expand these cells, they are specific for Aspergillus fumigatus and not to other antigens. SEB here is a polyclonal stimulator which we always use as a control, just to identify restimulated cells. And finally, it's pretty hard in humans actually to assess the in vivo regulatory mechanisms, but in vitro, the inhibition of proliferation of conventional T cells, activated T cells, is kind of a surrogate marker for the function of Tregs. And here you see the ability of Aspergillus fumigatus reactive regulatory T cells to inhibit the proliferation of fumigatus-specific conventional T cells very selectively. So according to all the criteria that are available, this is regulatory T cells. And now we go for the response in the conventional, in healthy donors first. And here we compare the Candida albicans specific and the fumigatus specific conventional T cells here. You see actually that all of us, or healthy donors, all of the cells are CD45RO positive. Most of them are CCR7 positive, so the classical central memory cells in the blood. For fumigatus, we have a different picture here. In a healthy donor, we have about 2/3 of the cells which are CD45RO low or negative, and we have about 1/3 of the cells which are CD45RO positive. Of those, most are CCR7 positive, but there's also some CCR7 negative cells. So these cells here probably are naive. These cells are pre-activated cells. Whether they are pre-activated by Aspergillus or by some other antigen are cross-reactive to Aspergillus, we cannot tell, because we have we don't know the history of these individuals. Now, if we go for these cells here, they don't produce effector cytokines, as is seen here. This is their phenotype, so they're kind of quotation marks naive cells. But if we go for these cells that are CD45RO positive in the healthy individuals, we can actually show that they recognize particular proteins of Aspergillus. This is a list of single proteins from the Aspergillus fumigatus lysate, or from the fungus. And you see here that particular proteins that are listed here with strange names, I don't want to read them, are actually recognized by these pre-activated cells of healthy individuals. Other proteins in blue here are not recognized. If we now go for the cells of an allergic individual, we see first of all that most, if not all of the cells are CD45RO positive, that is pre-activated. And these cells actually do not recognize the cells that a healthy individual recognizes. Instead, actually, they recognize another set of proteins of Aspergillus, which is here in blue. So there is, we call these Th2 targets because I don't show you the data. These cells are classical Th2 or allergic cells, and these is non-Th2 targets. So now the second part of the question is, so that was the conventional T cells. Now if we go for the CD137 positive cells, the presumptive regulatory T cells, we have here a healthy individual, and you see here that we have Tregs, the frequencies of Tregs that recognize the non-Th2, the green proteins of Aspergillus, and you also have ones that recognize the blue proteins, that is the allergenic or the Th2 targets. But if we go into an allergic patient here, we see that that person has actually regulatory T cells recognizing the green proteins, but it completely lacks regulatory T cells in the blood that recognize the Th2 the Th2 targets. So, and actually, if you plot it like this here for Z scores, you see that there's an exclusive thing. So the patients that have Th2 responses, so allergic immune responses to Aspergillus, don't have Tregs that are able to suppress or to recognize these particular proteins. So there's a divergent target specificity of fungus-specific regulatory Th2 cells and regulatory T cells and the Th2 cells. It's a lack of regulation, actually, in these patients. So I don't want to go much further into detail here. So the difference what the flow cytometry ARTID approach has shown for the first time, actually, is that in healthy persons there's a balance between naive T cells and regulatory T cells, which recognize all kinds of proteins from Aspergillus. In particular, the repertoire of the Tregs is complete. There is also a little population of probably cross-reactive, experienced conventional T cells already, but also these are kept in check by the regulatory T cells. So there's no, in healthy individuals, physiological immune regulation works out. While in allergic individuals, you have still Tregs that recognize some of the proteins of Aspergillus, but not the Tregs that recognize the same proteins that the Th2 cells recognize. So the Th2 cells kind of have evaded from immune regulation just because the repertoire of the Tregs is deficient. Now the last question is, what is actually happening in patients who have an invasive infection with Aspergillus? What's happening there? Do we have there also too many Tregs or too few Tregs? And how about the reactive T cells? And just one picture alluding on that, and what actually Petra and Alex have found, and that is that if you look in healthy donors versus two patients here with invasive infections, you see, first of all, you see that these patients have much less T cells, CD4 T cells, in their blood as compared to a healthy donor. It's about 1/10 actually of the frequency. And second, you see that actually the frequencies of Aspergillus-specific T cells is increased, at least in this patients also by a factor of about 10 as compared to this healthy control. So the total number of these cells remains the same. So there is fungus-reactive T cells in the patients. Why they are not able to control the invasion and the fungus is at this time not clear. But I think that cytometry will be a critical instrument actually to decipher to measure the reaction of these cells in the patients compared to the reactive potential of the cells from the healthy donor and find out in the end why is this immune reaction, which is there, apparently inappropriate. But already at this time, we can use flow cytometry to monitor reactive T cells and show that they are indicative for invasive fungus infection. So just to close up this part of my talk, so we think that cytometric identification of antigen-specific B and T lymphocytes according to reactive markers allows actually to address complex pathogens, mixtures of proteins, all the peptides of a protein independent of the MHC. It's a discovery driving thing, while I think that the direct labeling with the selected antigenic peptides actually for the T cells is good for routine, to translate this into routine use, but it will be very hard to discover the unexpected if you just ask for actually very defined repertoire of pathogens. We think that the enrichment of antigen-reactive T cells or artery technology lowers the threshold for detection, increases sensitivity, allows the identification of subpopulations and the analysis of larger volumes of blood. And so it just is actually the next step or a good way to approach lymphocytes reacting to a particular antigen. And finally, just a memento here that one of the critical things, of course, in this kind of approaches is the specificity controls. Now, how can we develop this technology further? And just one glimpse into ongoing experimentation is that actually we could actually go even further and analyze the antigenic potential of an individual by actually asking for the reaction to several antigens by barcoding the cells in the individual reactive slots. And that is, for example, showing here using two colors in different concentrations to label the CD4 cells in the antigen stimulation culture, and then actually having here, in this particular case, an assay of the CD40 ligand expressing cells versus interleukin-4 expressing cells, so it's Th2 cells reacting here against this antigen, that antigen, this antigen, and this antigen. So you can actually even increase this technology with more colors to get an immune profile of a patient. So the second point I want to address, after having talked about data that mostly actually are restricted to blood cells, is it actually all in the blood, the immune reaction? And just to introduce this part, a brief overview. It's actually a bit limited what we know about the numbers of different lymphocytes in the different areas of the body. We actually had to go to a 1974 publication in German to find any information on lymphocytes as such, not to speak of lymphocytes. That's actually a challenge for the community to find out how many memory versus naive versus regulatory T cells are in the various parts of the body. So what is available from this old publication, which actually was reviewed again 10 years ago, but that is it, yeah, is that in the blood we have something like 0.7 times 10 to the 10, so 7 billion lymphocytes. And that is just 1/4 of what we find in the spleen or in the bone marrow. Spleen and bone marrow have about the same numbers of lymphocytes. There's many lymphocytes, actually 10 times more in the blood than in the blood in organs like the gut, the liver, the skin, and the lung. And most of the lymphocytes actually are in the lymph nodes. This is 1.5 times 10 to the 11 of the lymphocytes are estimated to be in the lymph nodes. So actually there's pretty few of the lymphocytes are in the blood. And just for anticipating something that I will allude in a little bit more detail later on, if we go for memory CD4 positive or memory CD8 positive T cells, this is the relation of cells in the blood and in the bone marrow of humans. So blood is easy to analyze, but actually it's only giving a snapshot. And you don't actually know, are the cells constantly on the road, or are they actually on their way to leisure, death, work, or other work. So it's actually, you have a dynamic snapshot, but you don't really know about the state of the system in much detail. So we, some years ago, just asked a very simple, very, very simple question, and that was work by postdoc Koji Tokuyoda, where we tracked by cytometry memory CD4 cells generated in a defined immune response. This is one of these classical immunological transfer systems. You provide a Thy1.2 positive wild-type mouse with the T cells that have a T cell receptor specific for a virus peptide, LCMV peptide. You can discriminate the adoptively transferred cells because they express Thy1.1. You then immunize the mouse with the peptide and a very weak adjuvants, so because we want a fast immune reaction. And then we just follow up the fate of the Thy1.1, these cells here, cells which have been activated, so they start to upregulate CD44 expression in the various tissues. And that's what you get. So, I left out the tissues like the liver, skin, kidney, lung, because there were no cells, not in the beginning or not in the end. What I compare here is the lymph node, spleen, and bone marrow. So you see a typical immune reaction, fast expansion of the cell numbers, fast contraction within 3 weeks, 4 weeks. Then the contraction slows down, slows even more down. But after 3-4 months, actually there's very few, if any, cells in the spleen, and actually from the lymph nodes they disappear much before. What you see is actually that about 20% of the maximum cell number you find in the bone marrow after about 2 months. And then these cell numbers are actually kept constant. So if you look at the cells in the bone marrow and try to actually Paul Smith talked about these niches for tumor cells, but that's not a specific thing for tumor cells. Actually, also the normal lymphocytes actually need their environment to survive. And the nearest neighbor of the CD4 memory cell in the bone marrow is a VCAM1 positive, IL-7 positive stroma cell. So it's a particular subpopulation. About 40% of the stroma cells are able to express interleukin-7. It's known for a long time that memory is dependent on IL-7. It's not really clear how IL-7 works to maintain memory, but that's what we find. So nearly all of the cells, probably the rest to 100% is experimental failure, are actually contacting a VCAM1 positive, IL-7 positive cell. So a bone marrow survival niche for CD4 memory T cells. So the cells rest in terms of transcription. So they also rest actually, if I just briefly go back, in terms of mobility, because from this data here, you know actually that these cells are not circulating. So they are confined to the bone marrow. They are not found in any other compartment. That means they are immobile. And they're immobile, resting in these niches. They rest transcriptionally. This is comparing the genes differentially expressed between cells from the bone marrow and the spleen. And you see there's a highly interesting small group of genes here, which is upregulated in the bone marrow, downregulated in the spleen. But most of the genes are downregulated. Green is downregulated in this heat map as compared to cells of the same phenotype, CD44 high, from the spleen. So they're resting even more than the splenic cells. And they're also resting in terms of proliferation. For this, we actually use a marker called Ki67 in this particular very simple measurement. This is now, the previous data were mouse. I'm always mixing now mouse and human. This is human data. You see here the cells in the peripheral blood are about 3% for the CD4 cells, 4% for the CD8 cells, positive for Ki67, and the bone marrow is less than 1%. Interestingly, about half of the cells in the bone marrow, or for CD8 cells, even the majority express CD69. That had created a lot of confusion in the field because other groups have said these cells are pre-activated. But we know from previous work from Jason Sister's group and others actually, but Jason was, I think, the pioneer, that CD69 is not really an activation marker, it is a retention marker. It has to do with counteracting the chemokine receptor S1, sphingosine 1 phosphate receptor, and sphingosine 1 is actually the chemokine that lures cells into the blood. So the cells are going into the blood as a reaction if they express S1P. If they express CD69, they don't express S1P receptor. So, but that's just a sidekick and not really important for what I want to talk about here. So the rest in terms of proliferation. And Ki67 is a monoclonal antibody that was actually developed many years ago, 1983, by Hilmar Lemke, actually an old friend of mine, lab buddy. And it's actually recognizing a protein that is present during all active phases of the cell cycle. So Ki67 is present in G1, S, G2, and M phase, but the cells don't express it if they are in the G0 phase of cell cycle. So it means actually that the cells in the bone marrow are all in the G0 phase of resting in terms of proliferation. So that actually creates a problem for immunologists, because if you look up in the textbooks how memory cells are maintained, the mechanism that is described is by homeostatic proliferation. So actually the idea is that the cells, in particular in the bone marrow, are proliferating constantly and also dying constantly, and you have kind of a balance between dying and newly generated T cells. So why did we actually think that they would proliferate? And now comes something of technical relevance. So the statement that there is homeostatic proliferation in the bone marrow comes from addressing proliferation with brom desoxyuridine. Now, brom desoxyuridine, as shown here, has a brom atom here instead of this methyl group that is characteristic for thymidine. But the cells just incorporate BRDU into the DNA instead of thymidine, which is, at least if it is a limited amount, doesn't kill the cell. So, but it changes the DNA into a conformation that is recognizable by monoclonal antibodies. So there's antibodies that can discriminate between cells that have BRDU incorporated in the DNA versus those that have not. So it's taken up by cells synthesizing DNA and built and integrated into the DNA. That's actually the idea. And it's used to compare growth rates, the length of the cell cycle, or in pulse labeling, the history of proliferation of certain cells. Now, two groups, here Francesca Di Rosa, actually from Rome, and here another group, have actually used BRDU in different settings. And here was one setting actually where the mice have been fed with BRDU for 1 day. And then in the bone marrow, you see after 1 day, you see 7% of the cells BRDU positive. So in the Paretta publication, you wait for 14 days, and then you look up for BRDU incorporation here on the x-axis, and you see that about 60% of the cells, of the T cells you have incorporated in the bone marrow, have incorporated BRDU. If you extrapolate that, it means that every 2 months, every 1 month, the entire T cell population is divided once. Yeah? So we actually did a direct comparison head-to-head of Ki67 staining and BRDU staining. And we go here for mouse CD8 memory T cells, and we fed one group of the mice just got water. One got water with BRDU. Now, the technology asks, because the BRDU is bitter and mice don't drink so much of the water, so you add a bit of sugar. Yeah? So you also have here water with sugar, and here water with sugar and BRDU. And then actually you stain for Ki67 in the y-axis, BRDU in the x-axis, bone marrow versus spleen. And you see here actually that Ki67 positive cells are below 10% here. If the mice get water or water with sugar, it doesn't really matter a bit. Actually, we were wondering why this frequency was higher than the previous experiments, but it's still, it's 5%. But if you feed BRDU to the cells, to the mice, sorry, then you see that in the spleen, suddenly 30% of the cells, and the bone marrow, 70% of the cells become Ki67 positive. Or the clear-cut interpretation of this is that BRDU induces the proliferation of these cells. So it's an experimental artifact. We still don't know why or how BRDU induces memory T cells to proliferate, but it's definite that it does, actually. So next question is, so we have these cells resting in the bone marrow. We have cells circulating in the blood. What is the division of labor between these two memory cell populations? Or is it actually one population? And the cells are sometimes resting in the bone marrow and then at other times found in the blood. Actually, I already showed one piece of evidence that that the cells are not circulating, the ones in the blood, and that was from this artificial mouse system, this mouse transfer system, where after a certain while the cells recognizing the LCMV peptide were only in the bone marrow and not in the blood. You can also translate that to humans, and this is comparing now antigen-reactive cells for a systemic antigen that is all the time present, permanent antigen, tetanus toxoid here, then a group of viruses which are actually kind of historic antigens. Usually you encounter those as a child, or you get vaccinated against it as a child, and not much later on. And then two antigens which represent epithelial challenges, so vaccinia and candida yeast, which contact the body first and are limited to the epithelial surfaces. And what you see here is that just one example, one drastic example. So if CMV-specific cells you find both in the bone marrow and in the blood. This is actually what is plotted here. Here is plotted the frequencies of these cells, CD154 positive cells of individuals. That was people 30 to 60 years old who got a joint replacement operation. And so we were lucky to get a bit of the bone marrow of that person and also a little bit of the blood of that person. And every line is here connecting the frequencies of the cells in this individual's blood versus bone marrow. So you see the readily detectable CMV-specific cells in the blood, but actually the frequencies are higher in the bone marrow. Same for tetanus, with this exception. Probably this guy was vaccinated recently. For candida and vaccinia, actually the situation is complex, but for many people have more cells in the blood than in the bone marrow, and we would anticipate if we would have had access to the skin of these people, which we did not have, we would find even more. Actually, it's described in literature that the skin, our skin, is full of memory T cells reacting epithelial pathogens. Interestingly, the cells recognizing rubella, mumps, or measles, in particular measles here, are largely confined to the bone marrow. Actually, this is below in most probants, the concentration of measles reactive cells is below level of detection, very hard to detect. And you can even actually draw the line a little bit further. If you compare the phenotype of these cells in the blood and in the bone marrow of individuals, you see here in the open symbols the bone marrow and the closed ones the blood. You see that in the blood you find very few cells, and the very few ones express TNF, but no other cytokine. So it's lame ducks in a certain sense. While in the bone marrow you have many cells that express actually two of the three cytokines that we assay here, not only TNF but also IL-2 or gamma interferon, or even all three of these cytokines. A T cell type that has been called polyfunctional by our NIH friends, and they have actually provided evidence that this is the memory T cells that are protecting us from a challenge with these pathogens. So the bone marrow memory T cells are polyreactive, and actually can we mobilize them from the bone marrow into the blood? Yeah, so that is this experiment where we took somebody who had actually serum titers for measles, mumps, and rubella, and in Germany you are obliged to use a combination vaccine with all three vaccines, and so serum positive cells, which were very low for peripheral T cells, and we vaccinate that person, that donor, again. and then do our reaction stimulus. And you see here actually, before vaccination, we had 2 cells per milliliter reacting to rubella, so insignificant numbers. But actually, oops, sorry, that was the wrong, it's always the same thing here. But 16 hours after vaccination, we suddenly see a wave of cells jetting through the blood. They are there for about 1 day. After 2 days, these cells are completely gone from the blood. And then on day 3 to 7, they appear again. So the cells are mobilized from wherever. Of course, in this particular person, we don't know whether it's from the bone marrow or from other tissues. We can just guess that it is from the bone marrow. If it were from the bone marrow, it would be about 11% of the average content of bone marrow memory cells specific for rubella. And they are in there for just 1 day after the provocation, then go out, presumptively in the lymphoid organs, and then when the immune reaction goes on, some cells are released into the blood again for a little while. So I just show this actually to show you how important it is to go for the dynamic feature of cells in the blood. So it matters extremely whether you analyze these cells. Actually, we were surprised to find in literature nearly no data actually which the antigen reactive or antigen specific tetramer cells were measured just 1 day after antigenic provocation. Usually people go for several days, 7 days, 14 days, and so on. But you see already in this very narrow time range dramatic changes. So we think, just to sum up this part of my talk, that the resident memory T cells of the bone marrow maintain long-term polyfunctional memory to systemic antigens like viruses, measles, rubella, mumps. And we think that in essence to the immunological system immune system has developed a very deep actually defense system with the first line of defense already at the epithelia, the resident epithelial memory that other people work on. The second line of defense is a short-term memory of recently encountered antigens, and recently could be within the last few years in our lifetime. But if we go on decades, we have a third line of defense, which is sitting in the bone marrow resident memory cells, maintaining long-term memory in people that in their blood already are negative for memory T cells. So bone marrow provides protective memory to systemic challenges. Now, up to now, I talked about memory CD8 and CD4 T cells, and I will briefly now, towards the end of my talk, talk about memory plasma cells, and that is actually something that we work on since 20 years now. I think our initial, I'm not going into the history of this. This was completely cytometry driven. It's more kind of a historical talk, actually, of what kind of technologies we used to determine that plasma cells are long-lived memory cells by themselves, which was not believed at that time. Now, I just want to briefly actually take this up to show, because it has a clinical impact. And so plasma cells like the T cells are first generated in the spleen, and then later on you find them in the bone marrow. I can briefly go through that. They express 2 kinds of chemokine receptors, one sensing CXCL 9, 10, and 11, which is hallmarks of inflammation, one sensing CXCL 12, SDF1 alpha, which is a hallmark of several tissues, but in particular of the bone marrow. You can block actually the immigration to the bone marrow using a drug blocking CXCR4, AMD 3100. You see here complete block of immigration of the cells into the bone marrow. And you can actually, in humans, that's now my second example of how to monitor cytometrically immunity in the blood. You can actually monitor these cells on day 6 to 8. In this particular case, actually, the cells are labeled with tetanus. This person was immunized with tetanus, and we then go for CD27, and we gate on viable CD19 positive peripheral blood cells. And you see here actually CD27 positive and very brightly tetanus staining cells coming up on day 8. That is the memory B cells. But on day 6 to 8, you see cells expressing high amounts of CD27 and low amount, and staining very lowly with recombinant tetanus C. This is a wave of plasmablasts that flows through the bone, through the blood of humans, and is detectable exactly 1 week after immunization. Many other labs use that in the meantime to isolate the specific plasmablasts and the repertoire of the specific plasmablasts of those after immunization. And there's a lot of literature on that. Again, the timing is important. The plasmablasts are migratory, but actually this is a transfer migration assay. But if you, that is on 1 week after the immunization in mice. But if you wait already after 2 weeks, 12 days, the cells are completely resistant to these chemokines. They don't migrate in transfer migration assays. Instead, they still have the chemokine receptors, but they respond to it by survival. If you add CXCL12 to medium, isolated plasma cells plus medium, then you can increase the survival rate from below 5% to more than 50%. The plasma cells dock to VCAM1 positive CXCL12 positive cells, as shown by Rudi Manz and Anja Hauser some time ago. So we, and I don't show the data on the memory B cells, which are not yet published, which also are found in the bone marrow. So we think that the bone marrow is a particular tissue. Not only, it's like with salmons. So it's actually the tissue where the lymphocytes are generated from precursor cells and stem cells. But it's also tissue where they return to survive for long term in dedicated survival niches in kind of a dormancy, and as a resting cells in terms of proliferation, migration, and gene expression, activation. So that has implications for clinical implications now, this concept, because let's just look at a cohort of lupus patients, patients with systemic lupus erythematosus. And you can treat these patients with rituximab, cyclophosphamide, and glucocorticoids, and you will get 70% of reduction in IgG autoantibodies, double-stranded DNA antibodies. That's the antibodies that lead to kidney damage. So, and actually, in the end, to kidney failure, and make the lupus patients, they require transplantation, and this reduces the lifespan, et cetera, et cetera. 30%, we think, are from refractory memory plasma cells. And are these memory plasma cells pathogenic? Falkepe and Qingru Cheng just took plasma cells from the spleen of a lupus mouse model, NZBW mouse, and put those into cells that, to mice that don't have an immune system of their own, RAG-deficient mice. And actually, no, I need this. So here you see that the kidneys of these cells that received the plasma cells from the sick donor show all of the immune precipitates that the sick donor himself shows, and the rest of it is controls. Show that this is not happening with transfer of other cells. Now, how can you, is there any way how you can address these pathogenic plasma cells selectively? And I will just show how technologies develop. And this is this affinity matrix technology that we published in 1995, so 20 years ago. And Hyun-Dong talked about this. We used that for the identification and isolation of cytokine-secreting cells. Originally, we developed it for the isolation of antibody-secreting cells. And actually, we now go back to this technology, and we try actually to eliminate, to use it to eliminate plasma cells that secrete particular antibodies. So we have an affinity matrix that binds the antigen, in this case, ovalbumin, is bound to the surface of plasma cells with an antibody that recognizes syndecan-1, CD138. And so the plasma cell is labeled with the antigen that the antibodies recognize. And the antibodies secreted by this plasma cell are bound back to the surface of this plasma cell. And we think that there's complement around in vivo, and then the cell should be lysed. So does that work actually? Can we actually show this? And I don't go for all these controls. Just one summary slide, actually. This is what happens actually in mice, in which we have injected with this affinity matrix, and which have plasma cells specific for ovalbumin, and those for chicken gamma globulin, another antigen, just another protein. Yeah? And you see here the reduction in all the plasma cells is about 50%. There's no reduction in the chicken gamma globulin plasma cells. If you go for memory plasma cells, it's 75% reduction. If you go for, it was too fast. If you go for plasmablasts, it's about 50% here. So there is actually, you can use a cytometric technology that originally was developed to somehow label the cells or isolate the cells, also actually to, you can translate that into at least this experimental therapy at this time. So these plasma cells, we think, define a novel target of causative therapy. And the final 5 slides or so will be on actually, can we, if the blood is a very tricky tissue to use to monitor immune reactions and disease states, can we nevertheless use it as kind of a medium that indicates systemic changes in the immune state? And can we use it actually to monitor a systemic imprinting of the immune system, for example, by chronic inflammation? And this is work by Andreas Grützkau here and his group primarily. And the clinical problem behind that is the physical function, for example, in arthritis patients is going down by time. And actually, the earlier you treat these patients successfully, the more you can maintain the physical, the better you can maintain the physical function. But actually, there's a lot of choice, actually, how to, nowadays with these biologicals in rheumatology, we can use many different drugs targeting many different molecules or entertainers of inflammation. This is, each one is extremely expensive, so annual costs of one biological per patient is about 20,000 euro per patient. Yeah? And the tricky thing is that less than 60% of the patients, at most 60% of patients respond to a given biological. So 40% don't respond. You give the biological at all the expenses, but the physical impairment continues, the patients don't respond. So can we use cytometry actually to predict the response to therapy? And in our place, so Andreas actually developed this panel here. So we stain in 10 different tubes. We take 10 milliliters of blood, 1 milliliter each. We stain with this combination of things here. If you are interested in the details, that's published actually. And so we use up to 12 parameters and 7 dyes per staining, which all in all gives about 5,000 phenotypic parameters. And then we plot this actually in kind of a heat map. So this is not gene expression, this is immune phenotypes. Yeah? So this is marker combinations. And you see here 9 anti-TNF-alpha non-responders and 12 anti-TNF-alpha responders in a particular disease with reactive arthritis, yeah? Spondyloarthropathy. And you see here that you get a clear-cut difference in this heat map. It's a the clear clustering of the responders versus non-responders on week 0 of treatment. That is, you begin the treatment, before you begin the treatment, yeah, the immune phenotype is discriminated like this. You have 2 groups, actually, of patients. And if you go into the details, what is actually discriminating between non-responders and responders, you see here that it is monocytes a lot, it's eosinophils, to a lesser degree T cells, granulocytes, and NK cells. So it's kind of unexpected cells, not the antigen-specific cells. It's kind of granulocytes, leukocytes, innate lymphoid cells, which actually apparently have very sensitive sensors, very, very detailed sensors to take up the imprint of the disease and transport it to the blood and show it to us actually as a therapeutic measure. And that's what I wanted to talk about. This is just the people involved in this. The total, I mentioned them actually in passing by, the people who supported this, particularly the European Commission. And thanks to your attention.
Thank you, Andreas. Time for questions. And the micro, of course.
Andreas, not a technology question, but immunology question. So going back to the very beginning where you showed the homeostatic, well, the no homeostatic proliferation
Yeah.
of CD4 in the bone marrow. That was human data, correct?
Yeah.
I don't know if you also had mouse.
Yeah, we also have the mouse. I just
Okay.
had a choice, had to make a choice.
Yeah, so
what to show.
Yeah, the original data were from the oldest data were from Ralph Yamin, I think.
Yeah.
John Wary who did.
Yeah, yeah.
So
So
in that case,
I just took 2 publications
right.
for the BRDU thing. Yeah.
Right. So they use BRDU. So do you think that was an artifact of? Because they showed that there is a continuous homeostatic proliferation in the bone marrow.
Yeah, what I'm trying to say is
after infection.
I only had a chance here to show the Ki67 data, but we also have propylamidate data actually that show you whether the cells are in G1 or G0 versus cycling. Yeah, and it's the same thing actually. You can also show that BRDU induces in CD4 and CD8 cells, more prominently in CD8 cells, but also in CD4 cells. It induces proliferation. We think it is because of there's massive cell death. So because the entire thing is MyD88 dependent. So somehow there is antigen presentation. There is something going on, but we have not deciphered the mechanism so far. And to our knowledge, this claim of homeostatic proliferation is entirely based on BRDU.
Okay.
We are not aware of any propyl iodide or Ki67 or whatever alternative data.
And what about the in vivo labeling in humans? For example, deuterium labeling or heavy water and what was the
I'm
other
not
one?
aware
Carbon.
actually of any data showing homeostatic proliferation.
So but they
in
show
that situation.
They show turnover of the memory compartment.
I'm not so sure that what in the blood you have in the humans
Well, that's also the limitation.
1 to 2% of Ki67 positive cells. If you look at their phenotype, they express markers of recent activation.
Right.
So that is kind of not showing that in steady state. In the maintenance phase, the cells are proliferating.
Okay. Thanks.
So we are pretty sure about this claim.
People who work on DNA damage and repair use bromodeoxyuridine very carefully. It induces differentiation in myoblasts and a whole series of things.
Yeah.
And if you get electron abstraction from the BRDU, when it's in place, the cell undergoes a stress response and obviously undergoes DNA repair.
Yeah.
But of course, what you've done is introduce bromodeoxyuridine and completely skewed the nucleotide pool.
Yeah.
In fact, theoretically, what you should do with all bromodeoxyuridine or any nucleotide interventions is to balance the pools each time with the other 3
I mean, we are
elements. Yeah, so what happens, that's P53 dependent, exacerbated by reduction in P16 down the stream. For most situations, cells respond to that with the DNA repair response. But if they're suddenly being asked to do something metabolically, they start to mobilize their nucleotide pools. And so it's well known. And you're using it almost certainly, everybody does, at about, 5 millimolar? Something like that, 5 micromolar. I mean,
Yeah.
these are significant amounts for
Yeah,
a nucleotide,
exactly.
because what you're trying to do is saturate the pool to make sure you get a good signal.
Exactly.
In fact, it's the wrong thing to do. So
It's not us doing that.
Oh,
I mean,
yeah,
that
no,
was
I
the
understand.
previous publications.
No,
Yeah.
I
We have to
no, I
argue
understand.
with those people. Yeah.
Because I've basically done the same thing and regretted it, especially when you have DNA repair deficient disorders in the background, whether or not that's ataxic telangiectasia and also BRCA mutants as well.
Yeah.
So it is well known, but that's the limitation of the technique. And I think you put your finger on it exactly there.
Yeah.
No other questions? Well, thank you again, Andreas.